Hello student. In today's lecture we are going to talk about the diversity of cryptograms. So in previous lecture we have gone through the syllabus, and in today's lecture we are going to look towards the detailed concept which is included in this particular paper, that is diversity of cryptograms. So in previous lecture we have gone through the syllabus, that we know that the diversity of cryptograms is the first paper which is included in our syllabus, and morphology of angiosperm is the second paper, and based on these two different paper there will be a practical. So this is nothing but the total scenario related to the botany, and in accordance to this, another subject is there, that we can say skill enhanced subject is also included in our syllabus. So I am going to talk about the diversity of cryptograms in today's lecture. Now when we look at the unit number 1 of this paper, we can see that there is inclusion of viruses, general characteristics of viruses, classification, and as based on the economic importance, based on the host. Along with that, we are also going to concentrate on the structure, that is tobacco mosaic virus structure, and the multiplication of the same we are going to discuss in the unit number 1. Not only virus but also mycoplasma is also included in unit number 1, and related to the mycoplasma we are going to discuss the general characteristics of this mycoplasma. Then the third parameter which is included in syllabus, that is bacteria. In bacteria we are going to concentrate on the general structure, ultrastructure of bacteria, classification based on the shape, then reproduction and economic importance of the bacterial cell. These things we are going to discuss in unit number 1. Now the last point which is included in unit number 1 is cryptograms, that is general characteristics, classification. Then the next point which is included is lichen. For lichen we are going to concentrate on the general characters of lichen, nature or and association of the forms of thalli, then economic importance of lichen, structure and reproduction in Usnea. So Usnea is one of the example which we are going to consider for the lichen, and we are going to concentrate on this lichen. So in today's lecture we are going to talk about the viruses. Now as we know that viruses are the non-living structures, or we called it as a particles, which is present in nature, and they will get active when they will get the particular type of host. So they are obligate parasite, or we can say the viruses are the non-living particles which will get activate when they will get the proper type of host. So they are host specific, and that's why they are known as the obligate parasite. Viruses is neither an organism nor a cell. We considered virus as a particle, as they don't show any resemblance with the living organism or characters of the living organism. They are known as the uh, particles. They are known as the non-living structures. Now it consists of core of nucleic acid, that is DNA or RNA, and these DNA or RNA enclosed in an external mantle or covering known as or made up of protein. So protein is the external covering. The free state virus are quite inert. They remain as it is for the long duration until they will get a proper type of host. They become activated only when they infect the living host cell, and in the process only nucleic acid core enter the host cell, and it will start to multiply and produce the large number of cells of the viruses. The nucleic acid, which is genetic substance, takes over metabolic activity of the host cell and utilizes the cell machinery for the formation of more viruses. So in simple way we can say that viruses don't have a particular type of functional structures. They will utilize the host parameters and they will become the active, and they utilize the different parameters of the host cell. Thus, viruses are cellular parasites that cannot reproduce by itself. So, they require a particular type of reproductive material for their production. So, here this is nothing but the simple structure of the virus or viral structure. So, here you can see external envelope protein is there. Then again envelope is there, that is also made up of protein. Beyond that, inside to that you can see in viral integument is there, and inside to the parameter we call viral genome is present. The viral genome is either made up of DNA or RNA. One of them is there. Now, type of viruses. So, depending on their host, we can say that viruses are of different types. Say for example, here the bacteriophages are given, then adenovirus is given, then human immunodeficiency virus is there. Structures, when we compare, we can see there will be a lot of difference between this particular type of viruses. Not only these viruses are also known as host specific, and they will grow only in a particular type of host. For example, bacteriophages will grow only in a bacterial cell, they will not grow in an animal cell. Then human immunodeficiency virus, that is HIV virus, is particularly for the humans. It will cause the viral infection to the human only. So they are host specific, they cannot grow in a other host. Now in upcoming lecture we are going to concentrate on the classification of the viruses based on their host. So up till then, watch this video 2 times to understand the concept of viruses and take down the notes related to the viruses.